Now let's talk about the third chapter in general part, that is immunity. In the main exam, definitely the question is going to come, and the question will uh, range from 1 to 3. Now, what is immunity? We know native and adaptive immunity. So the native immunity is an immunity where you are having the immunity which is first line, and adaptive immunity is the second line of defense. Native immunity is always active, whereas adaptive immunity will require an antigen exposure. I told you to correlate this native immunity with your parents and adaptive immunity with friends. So memory specificity is not a part of native, whereas it is always present in adaptive. The duration of the lag time both are short in innate and both are long in adaptive. The components of adaptive immunity, they're very simple: B and T cells, the humoral and cell mediated respectively, and uh, the components of the native immunity, that's skin epithelial barrier, your dendritic cells, the antigen presenting cells, your WBCs, specifically neutrophils and macrophages, your complement products, C5a, C3b, and two more which which we will be talking about now. That's your PRR and NK cells, the pattern recognition receptors and natural killer cells. For a pattern recognition receptors, they are three in number, and they are having two patterns getting recognized. One is PAMP, the pathogen associated molecular pattern, and the second is DAM, the damage associated molecular pattern. Now, these pattern recognition receptors, PRR, they're three in number. One is in the cell membrane of the macrophage, second is in the cytoplasm of the macrophage, and third, it's going to be your endosomal receptors. Now, when the receptors are present in the plasma membrane, the receptors, important one, they are TLR and C-lectin receptors. The TLR, the toll-like receptors, they are, because they're the plasma membrane, they're outside the cell, so they can catch the extracellular microbe. The endosomal receptors, they're present in the endosome and they're also TLR. But the cytoplasmic receptors, one time a question has come on node receptor. What is node receptor? A receptor for necrosis and damage. Whereas RIG receptor, it is against the viral proteins. Now for TLR, just remember these types. TLR2 is against TB. TLR3 is against DNA RNA. 4 gram-negative bacteria. 5 F for 5, F for flagella. 6, 7, 8, they are for single stranded RNA, and 9, the mirror image, which you people tell us 6, but it is not 6, that's P, so protozoa. Okay. Then for NK cells, only 4 points to learn. देखो बेटा NK cell के नाम अपने को पता ही है, no, innate lymphoid cell, large granular lymphocyte, or a null cell. Why it is called as null cell? Because it is common ALL antigen negative, CD10 negative, which is positive for B and T cells. So it's a lymphocyte. So, there are three kinds of lymphocytes now: B, T, and NK. B cells 10 to 30%, T cells 60 to 70%, but NK cell are only 5 to 10%. Now, why we are talking about them? Because they're active against which cells? Any viral infected cell or tumor infected cell. This is the first point to learn. Second point, they cannot kill any normal cell which is containing MHC1. They're very powerful, but they will not kill anything which contains MHC1. Third point is the marker of NK cell: 16, 56, and 94. And fourth point, the interleukins for NK cell. If you want the increase in number of NK cell, you need interleukin 2 and 15. And if you want increase killing by NK cell, that is your interleukin 12. That's it. For this, the cells of the immune system, I told you there are three cells through: lymphocytes, the innate lymphoid cells, and the antigen presenting cells. Now, antigen presenting cells can be of two types: the professional, non-professional. अपन ने class में बहुत अच्छे से बात किया था that professional ones, they are containing MHC2, and non-professional one, that means they are containing MHC1. If something is containing MHC1, MHC1 is present on all the nucleated cells. So non-professional APC can be all the nucleated cells of our body. The only thing is that the antigen should be inside. So non-professional antigen presenting cell, they can only represent the antigen which is intracellular. Whereas professional APC can represent the antigen to the immune system even when the antigen is extracellular. That's all. The most important professional APC was the dendritic cell of yours. Okay. For a innate lymphoid cell, we just have read. For lymphocyte, there are B and T cells. B cell का क्या काम है छोटू? Antibody production. T cell, it can be of two types: CD4, CD8. CD8 का क्या काम है? Killing, cytotoxic T cell. CD4 helper है. तो क्या बोला था अपन ने? तीन टाइप का हेल्पर हो जाता है: मेमोरी, इफेक्टर, रेगुलेटरी. मोस्ट इंपॉर्टेंट सेल बताया था रेगुलेटरी, बिकॉज इट विल स्विच ऑन एंड ऑफ द इम्यून सिस्टम. जितना जरूरत है उतना ही. Memory मतलब memory बनाएगा, but effector cells which are going to produce the effect. So for production of different kinds of effects against different kind of antigens, which is the cell we have? Effector T cell. And how many types it is? Three types. TH1, TH2, TH17. कैसे याद किया था अपन ने बेटा? काला. TH1 is against chronic, TH2 is against allergen, and A is against acute antigens. The third one, TH17. Now TH1, it is 1. So it is going to secrete 1 plus 1, 2. So 2, 12, and gamma. TH2, it is 2 plus 2, 4, 4, 5, and 13. And TH17, it is 6, 8, 16, 22. Interleukin 2 is against CD8. Interleukin 12 and gamma, macrophage. 4, B cells. 5, eosinophils. 13, mucin. 6, 8, 16, 22, they are against neutrophils. ठीक है, तो ये अपना हो गया. Then we have the professional and the non-professional APC. मैंने आपको अभी बताया ये क्या है. B cells, they are 10 to 30% of total lymphocytes. The origin of it is bone marrow. The maturation organ is also bone marrow. अपन ने T cell के लिए बात किया था छोटू. T cell is also getting originated from the bone marrow. By why it is called as T cell? Because it is maturing in thymus. For a B cell, what is the function of a B cell? To catch the extracellular antigen because it's a professional APC. So it will catch the extracellular antigen, it will convert into a plasma cell and it will give you antibodies. So to catch the antigen, you need to have a receptor. So this is the diagram of the receptor. I told you the diagram of receptor. Head क्या है बेटा? IgM IgD chains. कान क्या है? Alpha beta. CR2 CD41. What is the action? Antigen binding. Signal transduction. This is the body. Production of antibody from here. CR2 co-receptor, it will help. CD40 co-stimulatory molecule, it will also help. What are the functions of these two? CD2, yeah, sorry, CR2 or CD21, इसका second option action क्या है? Entry of Epstein-Barr virus. And CD40, what is the second option? Attachment of the T cell happens at this by the help of CD40L. B cell can form antibodies. How many types of antibodies? Five. गामड़े GAMDE. इसमें से only antibody which B cell can make on its own is IgM, and the antibody which is formed, any other antibody than IgM, be it GADE, this all is formed how? With the help of T cell attaching with the B cell. Now the question which came in your exam this time is the molecule which is required for entry of EBV. So it was CD21 or CR2. ठीक है, तो ये देखो, this formation of any other antibody then IgM, ये कोई भी बनेगा. So the thing was that it is all called as isotype switching. And what is the interleukin required? Interleukin 4. याद रखना, interleukin 4 is required for isotype switching. T cells, again two types. Helper, cytotoxic. Helper ज्यादा होती है, cytotoxic कम है. What is the normal CD4 to CD8 ratio? 2:1. किस में कम होगा? HIV में किस में ज्यादा होगा? Sarcoidosis. Again, a T cell receptor. We discussed this, that for a T cell receptor, the antigen binding chains can be 4, alpha, beta, gamma, delta. Then co-receptor CD4, CD8, co-stimulatory molecule CD28, and the signal transaction molecule is CD3. B cell में you didn't need anything because the antigen could bind directly to the head because B cell was an APC. But if for a T cell you need to represent the antigen, you have to give it by the APC. So APC can be professional, can be non-professional. So APC will take the antigen. Which chains will bind on a T cell? Alpha, beta, gamma, delta. If the APC is professional, then alpha, beta chains will bind with the antigen. अगर APC professional है, professional APC, then this antigen binding will be alpha and beta. But if the APC is non-professional, then gamma and delta chains will bind. ठीक. अब how would you decide whether the APC is professional or non-professional? With the help of MHC. Because if there is MHC1, it's non-professional. Well, यहाँ पे क्या है? MHC2 है. So it's a professional APC. So antigen binding कहाँ पे होगी? Alpha and beta. Then this MHC2 bind with CD. They follow a rule of 8 that MHC into CD should be equal to 8. So if it is MHC, which CD molecule? CD4. And together it will cause 8. And one molecule was CD into CD. CD into CD, CD28 binding with CD80, 86. When all these three connection happens, then only the T cell will work. What is the most important molecule? CD28. Whether it's a B cell or a T cell, the most important molecule activation will be the co-stimulatory molecule. On a B cell, it was CD40. On a T cell, it is CD28. When you have all the three connections right, the T cell will function. Otherwise, not. So professional APCs, this is a professional APC because it contains MHC2. It will only activate CD4. And non-professional APC, that will only activate CD8. This is your T cells. Then hypersensitivity reaction, four types: acid, allergic, atopic, anaphylactic, immediate reaction, cytotoxic because of antibodies, immune complex mediated, and delayed because of T cells. Type 1 में देखो आपको याद क्या करना है? The examples of these hypersensitivities. But जैसे टाइप 1 है, इट्स एलर्जिक, फर्स्ट एक्सपोजर में क्या होगा? एंटीजन विल कम, एपीसी विल टेक इट बिकॉज इट्स एन एलर्जिक एंटीजन, सो TH2 सेल काला, CALA, किसके लिए? TH2. So it will be given to TH2 cell. 2 secretes what? 4, 5, 13. So all will increase. Interleukin 4 is responsible for what? Isotype switching. So your B cell will activate and will form allergic antibodies. Which are these? IgE antibody. These IgE antibody go and sit on the mast cell and sensitize the mast cell. That is happening in the first exposure. No clinical symptom present. Second exposure में IgE antibody attaches with the antigen, tells the mast cell antigen has come, let's act, and mast cell will release histamine. And what are the actions of histamine? Vasodilation, vascular permeability, contraction of smooth muscles. Immediate action within 2 hours of second exposure. And interleukin 5 and 13, they will increase mucin and eosinophil. So you will find 3 C's in the sputum of the patient: Crayola body, Kirschmann, Charcot. What are the examples? बेटा सारे allergic example है इसके अंदर. Important क्या है? ऑलवेज रिमेंबर दीज फोर, क्योंकि इनको एक्सप्लेन नहीं किया ना, इनको सिर्फ याद करना है. हे फीवर, प्रोस्नेट्स, कैसोनी, शुल्ट्सडेल, दे ऑल आर टाइप 1. बाकी कोई भी एलर्जी, फूड एलर्जी, ड्रग एलर्जी, एलर्जिक रिनाइटिस, एलर्जिक साइनोसाइटिस, एटोपिक डर्मेटाइटिस, ब्रोंकियल अस्थमा, ऑल दीज आर योर टाइप 1. टाइप 2, it's cytotoxic, antibody mediated. So इसमें antibodies, asking other cells to come and kill the cell. So this can be three mechanism: inflammatory, in which antibody binds with the normal cell of the body, makes it an antigen, and calls the WBC and kills it. Example was good pastures. Then phagocytic, in which antibody unnecessary binds with your blood cells, call the macrophage and kills it. Immune hemolytic anemia. Receptor dysfunction, either the antibody overactivate the receptor, Graves' disease, or antibody increase the rate of destruction of the receptor. So the receptor will not function. So again, it is myasthenia gravis. So in a receptor dysfunction, you have to understand it is not only decline in the function of the receptor. Sometimes increase in the function of the receptor is also a disease. And this receptor dysfunction of type 2 hypersensitivity is referred to as type 5 hypersensitivity. Okay. The mnemonic is: my blood group is Rh positive. Myasthenia gravis, blood transfusion reaction, good pasture syndrome, Graves' disease, immune hemolytic anemia, Rh disease of newborn, pemphigus vulgaris, pernicious anemia. My blood group is Rh positive. This is what you have to remember. And all the blood related hypersensitivities, they will always be type 2. Then comes immune complex. Beta, इसमें mnemonic याद रखो that antigen antibody, they bind and they destruct the body cell. Where they would be deposited? Wherever the filtration pressure is high, like kidneys and joints. The mnemonic is sharp. Sharp में क्या है? SLE, serum sickness, inoxazoline, arthurs, reactive arthritis, and PSGN. अभी PSGN क्या है? Post streptococcal glomerulonephritis. But I said in the class, all the glomerulonephritis are more commonly type 3 hypersensitivity, except good pastures, which is type 2. तो याद रखना, all blood related type 2, all glomerulonephritis more common type 3. Type 4, it is delayed, T cell mediated. So which TH cell will activate? TH1. Chronic antigen आएगा ना छोटू. So TH1 will activate and then it will secrete 212 gamma. 2 will increase CD8 cells, so more killing, and 12 and gamma, they will activate the macrophage, so granuloma formation. So always remember, granuloma formation is type 4 hypersensitivity. Examples: tuberculosis, multiple sclerosis, lepra type 1, contact dermatitis, type 1 diabetes mellitus. All these are your type 4. ये specifically I want you to remember this. ठीक है छोटू, एलर्जिक वर्ड आ रहा है बट स्टिल इट इज टाइप 4. लेप्रा में, लेप्रोसी के लिए देयर आर टू रिएक्शंस, टू, लेप्रा 1, लेप्रा 2. सो लेप्रा निमोनिक क्या है बेटा? ये मतलब व्हाट इज द फॉर्मूला? टाइप ऑफ लेप्रा प्लस हाइपर सेंसिटिविटी शुड बी इक्वल टू 5. So if it is lepra type 1, hypersensitivity should be 4, lepra type 2, hypersensitivity should be 3. So that the overall product comes to be 5. Now MHC, two types of MHC, 1 and 2. MHC3 is in between MHC 1 and 2 and it forms the complement products. 1 के लिए gene is ABC, 2 के लिए gene is DPDQDR, 1 के लिए chains are alpha 1, alpha 2, alpha 3 and beta 2, and MHC2 के लिए alpha 1, alpha 2, beta 1, beta 2. ठीक है, MHC1 is present on all nucleated cell plus platelets. MHC2 is present on all professional APCs. Your uses of MHCs are organ transplant and paternity testing. ठीक है. ग्राफ्ट रिजेक्शन, फोर काइंड्स ऑफ ग्राफ्ट, कई बार आ जाता है ये, इसको ध्यान से, आइसो आइडेंटिकल ट्विंस, ऑटो सेम इंडिविजुअल, एलो सेम स्पीशीज, एंड जीनो डिफरेंट स्पीशीज. Graft rejection, three types: hyperacute, acute, and chronic. Hyperacute is due to type 2 hypersensitivity within 48 hours. Type acute is type 2 more than type 4. This is within 6 months of grafts. And chronic, more than 6 months of graft, the transplantation, the graft is rejecting after 6 months of it, and it is delayed. So type 4 hypersensitivity. The cells responsible for graft rejection, याद रखना छोटू, it's T cells. Question आया तुम्हारे एग्जाम में. Graft versus host disease, the graft is more powerful, kill the person in which you have put it. It is seen after bone marrow transplant. It's type 4 hypersensitivity, also called as Rund's disease. Okay, this is graft rejection and GVHD. Amyloidosis, बेटा amyloid protein deposition, मोटी मोटी बात याद रखेंगे. There are systemic amyloidosis, localized amyloidosis, and familial amyloidosis. Now, when we are talking about localized, देखो सीधा सीधा amyloid plus disease. ACAL, a calcitonin है ना, so it is your medullary carcinoma thyroid. A beta Alzheimer's, A PR prions. Then we had AL, amyloid light chain, most common amyloidosis, primary amyloidosis, multiple myeloma. AA, secondary amyloidosis, seen with chronic diseases. Then familial में दो थे बेटा, FAP and FMF. FAP के साथ अपन ने क्या याद किया था, ATTR amyloid, and FMF के साथ AA amyloid. Most common stain is HNE, but the best stain for amyloid is Congo red. Gives you salmon pink color on light microscopy. That is why we have to do polarized microscopy to get apple green by refringence. Okay. Most common organ involved is kidney, A beta 2 amyloid with hemolysis. But most common cause of death, that's going to be your heart involvement. Okay. Now the biopsy which you are taking with amyloid deposition, most common site is abdominal fat aspirate, but the second best site is rectal biopsy. You can do electron microscopy. Electron microscopy will give you the diameter 7.5 to 10 nanometers and fibril structure. But X-ray crystallography, but this X-ray crystallography will give you the beta pleated sheet appearance of these. That's it. This is it for the amyloidosis. Okay. This you will, remember. इसको याद कर लेना, ये मैंने बताया तुमको. अदर डिजीजेस में बेटा ब्रूटन्स के लिए तो बात हो ही चुकी है, दैट यू डू नॉट हैव अ बी सेल एंड अ प्लाज्मा सेल बिकॉज बीटीके जीन इज डिफेक्टिव. For hyper IgM syndrome, the CD40 and CD40 ligand, this connection is weak. So only IgM is increased, rest everything is decreased. Then hyper IgE syndrome, STAT disease. Fate is the mnemonic, coarse facies, cold abscesses, teeth are absent in these, and eczema. Lot of IgE in these patients. Viscott-Aldrich, vast gene defect, mnemonic is water. And in ataxia telangiectasia, ATM gene mutation, and the patient will have VIA, the vasodilation, the ataxia, and immunodeficiency. So this is the chapter of immunity. The prime focus of this chapter would be on hypersensitivity reaction and graft rejections. Okay, so learn the mnemonic. I hope this quick revision of this chapter helps you in getting the concepts back and will give you the feeling that you can revise it. See you in the next. Thank you very much.